Hi everybody, welcome to another Chats with Chaudhry. I hope you're safe and well. Today I'm joined by Ellie Bickell, who is the assistant conference producer at Oxford Global for the Organ Modelling and 3D Cell Culture, which is coming up next week on March the 18th to 19th. So we are going to preview the event and find out what's going on at the event and why you should be attending if you've not registered yet. So Ellie, first of all, it's lovely to see you. How are you?
Lovely to see you too. I'm well, thank you. How are you?
I'm very well, thank you very much. Right now, first question for people not familiar with organ modeling and 3D cell culture as an event, what does it cover? And tell me a bit more about the event itself.
This is our third annual 3D cell culture and organ modeling congress. So the 3D cell culture part used to be part of our cell brand, but with the clear connections with the organ modeling field, this has since become part of Discovery and made up this congress. We're covering a range of the current areas in the field, including advances in drug development through the application of organ on chips and tissue on chips, as well as organoids and 3D cell culture, and how this is being researched within disease modeling, drug screening, tissue regeneration, and personalized medicine as well. Um, we'll have over 250 attendees from all parts of this field, creating quite a diverse audience, representing those key pharma and biotech companies alongside those academic institutions as well.
Cool. Now, I know having been to a recent organoids event myself only a few weeks ago, that organoids are a lot more in the news now than they used to be, and it's a growing part of research. So, what trends have you seen in this space when putting the program together?
So, one of the bigger trends I've seen recently is the focus on the development of an effective and standardized process to scale up both 3D cell culture and organoid models. So, successfully doing this may essentially revolutionize the field, as the evaluation of these models on a larger scale will help to reduce the use of animal testing and allow for the personalization of treatment across therapeutic areas. However, we first need to see this robust data to show the superiority of 3D and organ models to those animal models that are more widely used at the moment. Another key trend we've seen is, of course, the development of various organ and tissue on chips. So these are being used to mimic those in vitro organ and tissue structures. And these could be adopted into the preclinical drug screening of potential drug candidates, and if successful, would provide a much more accurate prediction of the toxicity, side effects, and treatment efficacy on human tissue and organs compared with those animal models. So those are two of the bigger trends I've seen at the moment, and these have been reflected in the design of the program to ensure that our content stays really cutting edge and relevant.
Well, that's such a beautiful segue. It's look like we planned this, Ellie, because my next question was going to be, what are the tracks that you've got this year?
So across the 2 days, we have three unique topic tracks. So starting on day one, these focus on organoid and tissue chip development, as well as the 3D model applications. So a couple of keynote presentations we have are from Stefan Platz, Senior Vice President at AstraZeneca, and he'll be covering the use of systems modeling to enhance the value of cellular models, and also from Martin Knight, who is a director at Queen Mary University, who will be reviewing the development of organ chip models in musculoskeletal tissues. Moving on to our second day, sessions will provide insight into the applications of organoid and tissue chip techniques alongside the trends in bioprinting and tools for 3D cell culture. So of particular excitement is our workshop entitled Creating Advanced Models Using 3D Bioprinting, where we'll hear talks from Nicola Corbett at MSD and Adam Leckenby at Mestag Therapeutics on the various applications and challenges associated with using 3D bioprinted models.
Great, fantastic. Now, apart from the tracks though, what else are you doing during the event to get delegates engaged and involved?
So we also have a variety of interactive sessions that delegates can get stuck into, dotted throughout the Congress. So our panel and roundtable discussions highlight the key current trends and challenges that we're seeing in the space. For example, we have a few covering overcoming the difficulties of using 3D cell culture model technologies, as well as the validation of organ models and organ chip models, how much complexity is enough. And this year, we're also hosting the Young Scientists Award for Best Poster Presentation. So this really celebrates the achievements of those early career scientists and highlights their contributions to the 3D cell and organ modeling field with the award ceremony at the end of day one. As well, we like to highlight that networking time during the breaks. So this is where attendees can connect with peers and relevant solution providers, really pioneering the 3D cell and organ modeling space. And at the end of day one, we also host a networking drinks reception where we encourage everyone to get involved in our speed networking session, which is always pretty fun to participate in.
Cool. So lots going on. Fantastic. Now, in terms of digital content, is the content available digitally concurrently or available on demand?
Yes, so whilst our events are fully in person, we do have our exclusive Plus Pass annual membership where those who would like to catch up on content after the event can. So this houses our on-demand presentations from all of our events, not just 3D cell and organ modeling, as well as providing full access to our content library and the opportunity to engage with the global community through participation in a range of webinars and online sessions as well. So this is a really great option for life science professionals to enjoy year-round exclusive insights, events and sessions with key opinion leaders so they can stay at the forefront of industry trends.
Cool, fantastic. Well, look, final question for you. Obviously, people listening to this and they might be interested in organoids and organ modeling and 3D cell culture, but they're still not convinced. Why should they be signing up for the event for next week?
So we've really curated our 3D cell culture and organ modeling congress to be a specialized hub for knowledge exchange, with the opportunity to not only hear from and connect with those key opinion leaders, but also to explore the latest advancements in the technology being used to completely revolutionize the way we develop treatments for patients. So if you are at all curious about 3D cell culture or organ modeling, this is the place to be. And it's happening next week, so don't miss out.
All right, fantastic, Ellie. Well, thank you for that. Well, there you go, viewers. If you're involved in organ modeling and 3D cell culture, or you're not involved, but you'd like to learn more, then click on the link above the video and learn more. And hopefully I will see you there next week for the event as well. So, Ellie, good luck with the build-up. Viewers, thank you for watching. Until next time, as always, stay well and stay safe. Bye-bye.